For today, I want to follow on from the stuff that we were talking about yesterday and talk about the three major cortices in the mammalian brain, being the cerebellum, the cerebral cortex, and the hippocampus. So what I want to do is yesterday we were talking about things in a very esoteric neuroscience sort of a way. What I want to do now is to come down and pick up particularly on the relationship between structure and function. I introduced a couple of concepts about the ways that networks worked and the ways that they were put together, and I want to look at those in more detail today and just use these three examples as different ways in which neural networks can fit together. So we started off with a small world network. And you'll remember that this is a network where each cell is very likely to be connected to its near neighbours, but very unlikely to be directly connected to a neuron some distance away. And I highlighted the advantages and disadvantages of this network. The advantage being that it, relatively speaking, consumes less energy because it has a shorter length of axon per neuron than the other networks, and the disadvantage being that it's not good at talking across the whole of the network. But, and this is another advantage, within small sections of the network, it's very good at processing information quickly. We said that the cerebellum looks like that. This is the sagittal section of a dog's brain, and I have highlighted the main areas of the cerebellum. The cerebellum is divided into the rostral and caudal cerebellar cortex, or anterior and posterior if you're human. The terminology there is different because of the anatomical differences between those two brains, and also the deep cerebellar nuclei. And what I want to do now is to have a look at the way that the cerebellar cortex is built at a cellular level and just show you how those cells fit together to serve that local coordination function. The first thing to say is that the cerebellum has lots of inputs and very few outputs. And depending on species, the ratio of inputs to outputs is something like 40:1. So for every axon that comes out from the cerebellum, there are 40 axons going into the cerebellum. The cerebellum is very much a watcher within the central nervous system. It monitors all sorts of information to see how things are going. It knows about lots of things that are going on in different parts of the central nervous system. And its limited outputs tend to plan for motor function and to coordinate motor function so that motor function is smoothed out. Given that we look on the cerebellum as a smoother and a coordinator of motor function, it's quite interesting to note that if you sit and deliberately decide to do a motor function, then the cerebellum is the first area of the central nervous system that starts to undergo electrical changes. So if one of you decide that you want to pick up your drinks bottle and take a drink, then before your hand moves to the drinks bottle, the activity will occur in your motor cortex and in your non-pyramidal motor system, which allows that to happen. But before that happens, the cerebellum will be planning and getting ready for that activity. And electrical changes in the cerebellum can occur usually about 2 seconds before any volitional motor activity. That's another example of your left cerebral hemisphere convincing itself that it controls the world when it really doesn't. Okay? Your perception of reaching to pick something up and picking it up is that I decided to reach over and pick it up and then I did it. But actually, you can only decide to reach over and pick something up when your cerebellum tells you it's good and ready to allow you to do that. The cerebellum does all the planning and gets all the coordinated motor function in place and adjusts posture so that you don't off-balance yourself by doing whatever it is you want to do. And then it says to the left cerebral hemisphere, okay, good to go now, we can do that. And the left cerebral hemisphere says, ooh, that's cunning because I just decided to do it anyway. And gives you that impression that you're in control. Whereas the actual truth of conscious control is a more distributed function throughout the central nervous system. So the inputs, most of them go to inhibitory interneurons, and there are many. Some of them go directly to the deep cerebellar nuclei, and some of them go to the Purkinje cells, and the Purkinje cells are the only output cells from the cerebellum via the deep cerebellar nuclei. And if we look at the regional anatomy of the cerebellar cortex, you can see that different parts of the cerebellum deal with different processes. This is effectively data taken from the cat. So this is the cerebellar cortex of the cat, dissected off and kind of squashed flat. A bit like a Mercator projection of the Earth is the Earth kind of unpeeled and squashed. And when you do that, you see that these different areas of the surface of the cerebellum deal with different functions. And so this large green area deals with the pontocerebellar activity. It's called the pontocerebellum. And it communicates with neurons in the pons, in the brainstem, which are largely upper motor neurons, principally of the extrapyramidal motor system. And this part of the cerebellum is planning for and adjusting motor activity at the level of the brainstem. The spinocerebellum, in blue, communicates with the spinocerebellar tract, which travels down the spinal cord and communicates with the segments of the spinal cord, and deals with such things as segmental posture, and collects information about the posture of the body and the positions of the limbs in order to keep an overall picture of postural integrity, and also sends out information adjusting posture. And finally, the vestibulocerebellum deals with the vestibular system and coordinates the sense of balance. And all those activities together are dealt with over the surface of the cerebellum. When we look at the cerebellar microstructure, we find that the grey matter of the cerebellum has 3 layers. So these peripheral 3 layers are all layers of grey matter. And this 4th layer here is the white matter that underlies that. So those are the tracts that carry information going to the cerebellum and leaving the cerebellum. The layers we've got are the so-called molecular layer peripherally, the Purkinje cell layer in the middle, which houses the cell bodies of the Purkinje cells, and the granular layer more deeply. So if we look at the cells, you can see that the Purkinje cells, these red cells here, have their cell bodies in the Purkinje cell layer. And you'll remember, hopefully, that probably in the 4th or so lecture that I gave you, we looked at Purkinje cells as an example of a multipolar neuron, and we talked about this large dendritic tree being 2-dimensional, and that's represented here by this Purkinje cell in this section having a wide dendritic tree, and this Purkinje cell having a very narrow dendritic tree. Both trees are kind of facing in the same direction, like slices of toast. Notice also that different neurons tend to impinge on the Purkinje cell dendritic processes in different ways. So that the basket cells tend to synapse with the Purkinje cells around the cell body, and they tend to encase the Purkinje cell in a basket of axonal processes. The stellate cells are much more peripheral, and they tend to impinge on the apices of the dendritic processes in a much more peripheral manner. There are two sorts of fibres climbing into the molecular layer, but we'll get there in a minute. And we've got these granule cells which climb up superficially. Now, the granule cells are interesting because I've said that the dendritic processes of the Purkinje cells are lined up like slices of toast against each other. The granule cells are the cells which run at 90 degrees to those slices, and so the granule cells will have axonal processes on a number of different slices of cerebellum, and they're the ones that carry the long distance information. But they are relatively sparse compared to all these basket cells and Golgi cells and stellate cells that have very short axonal processes and deal with information very locally. So we've got a picture here of a cortex which has a very intricate local level of organisation with a very sparse level of long-term communication running through it. If we look at those axons, we've got the Purkinje cell axons leaving the cerebellum and going to the deep cerebellar nuclei and then outwards. We've got mossy fibres and climbing fibres coming into the cerebellum, and those mossy fibres and climbing fibres synapse in different ways with the cells of the cerebellum. So that the mossy fibres synapse with granule cells and tend to contribute to this long distance innervation, and the climbing fibres deal much more directly with single Purkinje cells, and so they have very arborized axonal processes that match up with these dendritic processes of the Purkinje cells. It's also not very well illustrated on that previous slide, but better illustrated here, that the Golgi cells are feeding down into this granule cell network. So the Golgi cells are sampling information from these fibres, both the dendritic processes of the Purkinje cells and the dendritic processes of the parallel fibres, and feeding that information back down into the granule cell network. And so we've got these very localized loops of information processing going on with smaller contributions from elsewhere. It's a very localized system. And it deals with things very rapidly. If you have a postural imbalance so that you're about to fall over, it's no good taking a third of a second to deal with that, because by that time you'll be on the floor picking yourself up. And so we need these very rapid response times that we get with very localized signaling systems or neuronal systems. And the price we pay for that is the same price as the advantage, the fact that they are localized and so they don't deal particularly well with information from distant parts of the body. Going back to those climbing fibres and mossy fibres, they are different from each other also in the kinds of action potential information that they carry. So that the mossy fibre tends to carry information as single action potentials, whereas the climbing fibre tends to carry information as bursts of action potentials, several action potentials together. And so those fibres are both affecting that cerebellar network in different ways from each other. Let's contrast that now with the intermediate network. You'll remember that the intermediate network has many local short communications, but also a number of longer distance communications which travel to different parts of the network. And so, although it's still able to locally process information, it's beginning to get into a situation where there's a more global look at the information, a more global view of what's going on. Here is a diagram of a human cerebral cortex, and you can see that we've got lots of different areas, many of which you'll be familiar with, which are processing different sorts of information. So here, in the caudal part of the brain, we're dealing with visual association in the optic cortex. As we go more frontally, we're dealing with sensory information and then motor information. And as we go into the premotor area and the prefrontal cortex, we're dealing more with higher order thinking and cognition and so on. Notice that even though these localized areas are localized so that they can process information rapidly within that area, there is obviously going to be communication between those areas to carry out overall function. So that, for example, when we are listening to speech and responding, Wernicke's area here of sensory speech has got to be communicating with Broca's motor speech area, and the whole thing is taking place over the whole surface of the cortex. But it's taking place over the surface in a localized way. Different bits of cortex are dealing with different packets of information. And we can see that localization if we look at the somatotopic localization in the sensory cortex. This is a mouse, and the whisker pattern on the mouse's cheek has been highlighted here with black pen. So every whisker has been highlighted at the point where it goes into the mouse's cheek to give you this pattern of black dots. Now, if we look at the mouse's primary sensory cortex, we get a homunculus, a representation of the body in the sensory area, and you can see that the representation of the whiskers in a spatial sense in the cortex is the same as the spatial arrangement of the whiskers in real space. If we then take a slice through that cortex, parallel to the surface of the cortex, and look at a histological section, you can see that we've got areas where we've got groups of neurons with areas where there are neuronal pathways between them, so that the cell bodies are dense in these areas, but these areas between them are sparse of cell bodies and contain axons which connect the bodies. And also, I hope you can see that the layout of areas within this part of the cortex is the same as the layout of the whiskers in real space. It's an example of somatotopic localization. The topic arrangement of the central nervous system looks like the topic arrangement of the soma, the body. What I want to do now is to take a slice not parallel with the surface, but to take a slice perpendicular to the surface and look at what happens as we go down the cerebral cortex. This is a slice of cerebral cortex which is stained different colours for different neurons and their projections. And you can see that these areas where there are increased numbers of cell bodies go down in columns through the entire depth of the cerebral grey matter. There are lots of intricate connections between cells in the same barrel, or column as we call them. And between barrels, there is a sparser connection of longer fibres. Yeah, so we've got this dense local interconnection of neurons, but we've also developed longer neurons that are carrying information between processing units. And that's reflected at all scales within the cerebral cortex, so that some of these long fibres will be going to distant areas of the cortex, and some of them will be going through the corpus callosum to the opposite cerebral cortex. So the density of connection is present over long distances, but is much more dense over short distances. And that gives us this functional unit concept of processing of information and dealing with motor function. That harks right back to Kajal's initial diagrams of the cerebral cortex with its layers, and means that we can divide the cerebral cortex into not 3 this time, but 6 layers, where we find typical examples of different cell bodies. But in essence, the pyramidal cells in the cerebral cortex are the cells that are the origin of those long fibres that travel to distant places, just like the Purkinje cells in the cerebellum. The last network to look at is the random network, where any neuron in the network is just as likely to be connected to a neuron on the distant far side of the network as it is likely to be connected to its near neighbour. The connections are entirely random within the whole structure, and we don't see any of those structural regularities. The hippocampus looks like that. This is a section of the hippocampus, again stained for different sorts of neurons, and you can see that we have layers of neurons across the hippocampus with no real distinction between them. They all kind of flow into each other in one big amorphous layer that doesn't have areas within it that are easy to identify. I've shown you this diagram a number of times now, with a hippocampal neuron that has local connections and also distant connections. And if we look at a real hippocampal neuron, this slice of hippocampus has one particular neuron stained in this light yellow colour. And you can see that this particular neuron, who knows where its cell body might be, has got projections pretty much reaching all over the hippocampus. The whole thing is one amorphous structure of neurons that are hugely interconnected and are speaking to each other across the whole structure. The hippocampus tends to be the scratch plate where we do our short-term processing. It deals with short-term memory, it deals with short-term spatial regulation, it deals with things that come and go. And it's a very flexible, high-maintenance network that allows us to deal with information in a very flexible way. So I want to look now at some of the processes that go on in those neurons of the hippocampus that allow us to process memory and learning. First thing to say is that there are 2 kinds of memory. There's explicit and implicit memory. Explicit memory is a conscious recollection of something. And implicit memory is more an effect on your thought process or an effect on your motor activity. And I guess the usual test that's used to decide whether a memory is explicit or implicit is, can you directly relate it to your neighbour in language and get the whole of the concept of the memory over? So, for example, those of you that are making notes at the moment will be writing letters down, and I guess the letter A might be one of those letters that you're writing as you take notes down. Can anybody remember when they learnt to write the letter A? It's not something that you have an explicit memory of, but it's something that you have developed over time as a motor skill, so it's an implicit memory. And also, if you take somebody who doesn't know how to write a letter A, and you sit there with your hands under your legs and just explain to them using language how to write a letter A, it's not going to help them very much in making a really nice, neat letter A. It's a motor skill that they need to get for themselves. Whereas if you have something that happened to you in the pub last night, you can tell the person sitting next to you all about it, and you can get the whole of that concept over using language. Being English, I know more than I would really like to admit about the Battle of Hastings in 1066, and because I studied this at school, I can tell you that older sources tend to say that this is King Harold getting his arrow in the eye fairly famously at Hastings, but that actually the Norman people that built the Bayeux Tapestry or sewed the Bayeux Tapestry tended to indicate with script an event that was happening at the end of script. So where it says King Harold is dead, Harold Rex interfectus est, it's not really talking about this chap here that's getting the arrow storm, it's talking about this chap here that's getting it with a sword from a Norman knight. And so the whole thing is a bit of a historical misinformation. And I can tell you that because I learnt it in school, and I can convey that to you with language. It's an explicit memory. Who can play a musical instrument here? What do you play? Piano. And you don't play a musical instrument. Okay, if I sat you down in front of a piano and got you to tell her which keys to press and how to make a nice piece of music, how would it sound? Not very good. So you couldn't convey that information over with language. It's a motor skill that you've learned probably from countless hours of tedious practice working out how to do that and just kind of getting it into your motor system. So it's an implicit memory. You have this motor knowledge, this memory of how to play the piano, but it's not something that you can relate to a specific incident or teach somebody just through language. And things that are implicit tend to be motor function, attitudes, phobias, and emotions. Yeah? If you don't like spiders, for example, that's probably something that you weren't born with, it's something that you've learnt. But it's not something that you've probably learnt through a specific incident, unless, like Billy Connolly, you met one in an Aussie toilet one day and decided that you weren't going to like them forever. But for most of us who don't like spiders, it's something that we've kind of developed as we've grown up, and we can't hark back to the incident that made us not like them. It's something that it just becomes part of us. If we talk about the mechanisms of learning, there are three major mechanisms: habituation, synaptic development, and long-term potentiation. And I want to talk to each of those now. The first two using the example of aplysia. Aplysia. The humble sea slug. The reason we're talking about it in the sea slug is that most of the work looking at these kinds of memory were done with sea slugs. And the reason the work was largely done with sea slugs is because the average sea slug has about 27 neurons in it. And so we can model that and we can more or less work out what's going on. If we start moving to mammals and we get 6 billion neurons, things are a bit more difficult. So with a model of 27 neurons or so, we can more or less work out what's going on. So let's look at habituation, first of all. Habituation says that we get used to things. We get habituated. So, here is a sensory neuron in the sea slug being stimulated, and here is a motor neuron undergoing an action potential and undergoing activity. And what's happening in this model is the sea slug has two structures on its back here. It has a siphon, which is a tough structure, which pushes fluid out, it pushes seawater out so that it can filter the seawater and get food out somewhere down in its bowels. And it has a gill, which it uses for respiration, which is a very delicate and very vascular structure. And if you touch its siphon, its gill goes, ooh, something happened, and pulls itself in as a protective mechanism. So here we've got a sensory neuron being stimulated by somebody touching the siphon, probably with a small paintbrush in the experimental studies, and a motor neuron sending out an action potential to contract the gill to get it out of the way in case the paintbrush should come looking for it. If we repeat the stimulation again and again and again, we find that we keep the sensory information coming, but the motor neuron stops sending motor information. The animal becomes habituated. It gets used to the idea that people are always touching its siphon with a paintbrush. It's not a big deal. The world isn't about to end. We don't have to contract our gill and stop breathing for a while. And if we habituate it, you can see that at 1 week, we've retained that habituation. There's still no motor response to that sensory stimulus. And if we look at the percentage of detectable connections within that nervous system when it's habituated, compared to control here, you can see that at 1 day after habituation, we're down to something like 20% of connections, maintained for a week, and at 3 weeks, it's still only beginning to recover to the control value. So we've got a system where we can alter the way the system works, and that alteration will stay for a considerable period of time. If we look at the reverse, we look at sensitization, we can also see that that happens for a period of time. So what we've got is sensory neurons in the skin, on the siphon, and in the tail, which are feeding into facilitatory interneurons, which are feeding out to those motor neurons which are causing the gill to contract. And if we sensitize these skin areas, either by not giving a stimulation for a long period of time, or by giving a very strong noxious stimulation, then the next time we stimulate this area, we get a very large contraction of the gill. The snail really thinks something exciting is about to happen, and it pulls its gill well out of the way to protect it. So here you can see that the sensory neuron has the same stimulus, but the motor neuron this time has an increased stimulus. If we look at the mechanisms by which that happens, we find there are 2 sorts of mechanisms in place. The first one is governed by the amount of neurotransmitter that is secreted by those sensory neurons and interneurons. So in the control animal, we stimulate the siphon and we get the release of a goodly amount of neurotransmitter. But in the habituated animal, that stimulation of the siphon leads to the release of less neurotransmitter. So the animal's neurons are responding less to that stimulation in a chemical sense because they're habituated. We also, with time, get changes in neuronal development, so that if this is the control model, with the interneuron having 2 connections onto the axon, in the sense of chronic habituation, we get a reduction in the number of synaptic connections, and in the same case of chronic sensitization, we get an increase in the number of connections. So we've got 2 changes there which are complementary. Each synapse is increasing or decreasing the amount of neurotransmitter that it produces, and the number of synapses is also decreasing and increasing, changing the hardwiring of the nervous system. And that harks back to a slide that I showed you in the vision lecture when we were talking about the degree of connectivity in the optic cortex of the mammal. That sensory information causes physical changes to occur within the central nervous system, which then get hardwired in place. And so those changes persist into the future. The final mechanism for memory is a mechanism called long-term potentiation. And it's a mechanism whereby the resting membrane potential of those neurons can change in the long term, so that they will sit at rest either closer to threshold and be sensitized, more likely to be stimulated, or further away from threshold, be hyperpolarized and desensitized or habituated. And here we've got an amplitude of a postsynaptic, excitatory postsynaptic potential in control animals compared to animals which are given a number of conditioning stimuli. And you can see that with each conditioning stimulus in the stimulus group, the magnitude of the excitatory postsynaptic potential gets larger because the resting potential of the membrane is changing. It's hypopolarizing, it's becoming more sensitive, and so these EPSPs are having a greater effect on it, compared to the control group, which just ticks along. This study was done in mice looking at learning of an electrical noxious stimulus. The workers were interested to see how long this postsynaptic potentiation persisted, but they weren't able to find out in mice because the mice die before it goes away. The postsynaptic potentiation lasts for longer than the mouse lasts for. It can be an extremely long-term feature of the nervous system. But it can also be turned off very quickly and used as a short-term store of information. So let's look at a different mouse. And let's put this mouse into what's known as a Morris maze. A Morris maze is a particularly unpleasant way to tease mice. It is a large circular area of water which is just deep enough so that the mouse can't put its feet down and it has to swim around in it. In one area of the maze, a little platform is placed so that it's still under the surface of the water so that the mouse can't see it, but when it gets to that area, it can sit on the platform and it can rest and still breathe. When you put a mouse into this maze, it swims round and round and round and round and round, and then it finds the platform and it goes and sits on the platform for the rest of its time. If you put orientation cues around the maze so that the mouse can see more or less where it is in the maze, so that here it's near the triangle, here it's near the crescent, here it's near the hexagon and so on, and you always put the platform in the same part of the maze, the mouse can learn where the platform is. So that when you put a mouse that's been in a maze for 10 times previously into a maze, it will beetle over straight to the part of the maze where the platform is and swim around till it finds it and sit in it. It learns to deal with the maze much more efficiently than a mouse that hasn't had previous exposure. And we do that by long-term potentiation. When we go into an environment that we are familiar with, we load a mental picture of that environment into our hippocampus by altering the potentiation of different cells in the hippocampus. And we all do that with most of the environments that we go in. So in this lecture theatre, are you sitting today in more or less the same place that you usually sit when you're in this lecture theatre? Yep, most people will be, most of the time. And that's because when you come in, you don't think, right, I'll sit here at random and I'll learn this new environment. You come in and you say, ICLT, I've got a mental map of that, pop it into my hippocampus, and this is the bit I feel comfortable in.
The way we do that in the hippocampus is through chloride channels, NMDA channels.
We can demonstrate that by making mice that don't express those hippocampal channels in the hippocampal neurons. And if we put one of those mice into a Morris maze, doesn't matter how many times we put it into the Morris maze, it just swims round and round and round at random until it happens to find that platform. It's got no strategy for learning where the platform is, and it doesn't get any better at looking at where the platform is. So that ability to learn our way around a new environment is a feature of the hippocampus, and it's a feature of having that ill-defined, flexible learning space that we can randomly feed information into. In terms of the longer term LTP, it may well be that a feature of the shape changes of dendritic spines that I talked about quite near the start of the lecture course is what causes those alterations in long-term potentiation to be so well maintained. If you think about it, receptors work in the milliseconds range. And it seems kind of unreasonable to expect a system that works over milliseconds to still be able to work about a year and a half later. And that was some of the longer-term LTP studies from that mouse experiment. So it may well be that NMDA channels within these dendritic spines are causing a change in shape of the dendritic spine and altering the functioning of that neuron's dendritic tree at that point. And so contributing to the changes that we get in membrane potential in the neuron. And they may be more or less flexible over time depending on the age at which those changes were first made and depending on the kind of neuron that made those changes. And they can affect all sorts of things. So going right back to one of the first lectures where we talked about properties of integrating centres, most of these properties can be altered by learning in some respect. So you can learn particular skills that will enhance your reaction time and shorten your reflexes. You can learn feats of memory and you can learn how to store more and more information in your short-term memory. Most people can store between 7 and 9 random numbers in their short-term memory. So if I give you probably 9 numbers, you could give them back to me. But if I give you 10 numbers and ask for them back, you're going to start to get a bit confused and they're going to get muddled together. But by practicing and working through those systems, you can increase the number of digits that you can maintain in your short-term memory at any one time, should you want to do that. So all of those things can be changed in different ways by practicing and by making our central nervous systems do those things that we want it to get better at.